Concerning the live virus vaccines, how are these vaccines produced? They consist of attenuated, harmless live viruses capable of triggering an immune response without being harmful. How can we attenuate live viruses? The live viruses are engineered by removing from their DNA the harmful genes that are responsible for the disease and replacing the harmful genes with non harmful ones so that the viruses are harmless but can still trigger an immune response. In this way, we can attenuate the virus and make the live virus vaccine. Now, we will see an example application on live virus vaccines concerning the herpes disease. How can we produce a vaccine against herpes disease? To remember, the immune response triggered against the live virus vaccines or the peptide vaccines is characterized by the production of antibodies by B lymphocytes. The antibodies are specific for the envelope protein of the virus and not for the whole virus. Concerning herpes disease, herpes is a disease caused by a virus called HSV virus. This disease is characterized by skin lesions, eye lesions, genital ulcerations, and fatal encephalitis. Once infected by this virus, the organism develops a specific immune response characterized by the secretion of antibodies against envelope proteins of the HSV virus. So the questions can be, for example, pick out from the text the reaction of the organism against HSV. You have to pick out the whole sentence from here. Once infected by this virus, the organism develops a specific immune response, secreting antibodies against envelope proteins of the HSV virus. Now pick out the symptoms of herpes disease. The symptoms of herpes disease are 2 points, and you list the symptoms: skin lesions, eye lesions, genital ulcerations, and fatal encephalitis. Now, in the second question, describe the technique of producing a live virus vaccine against the herpes virus. So a biotechnological, a DNA recombinant method, a classical method permits the production of a vaccine against the harmful herpes virus using another virus which is harmless. So the live virus vaccines are produced by using 2 viruses. The first virus is the harmful virus against which we make the vaccine, and we aim to produce an immune or trigger an immune response. And we use another virus which is harmless and which will be injected into the body and which is a vector that will transfer the genes from the harmful virus into our body in order to trigger an immune response. So as you can see in this document, the first step is to extract the gene or the DNA from the herpes virus. So we extract first the DNA from the herpes virus. We need to isolate the gene that codes for an envelope protein of the virus. So in order to isolate this gene, we use restriction enzyme to cut the DNA and to isolate our interest or our gene of interest. Now, from the harmless virus, we extract and cleave the DNA. Then we integrate the herpes gene into the DNA extracted from the harmless virus. So we obtain a recombinant viral DNA, a DNA from a harmless virus carrying a gene from a harmful virus, which is the herpes virus. The recombinant DNA obtained is reintroduced into the harmless virus. Now, the recombinant harmless virus is the vaccine and it is injected into the body. Now, the envelope protein of the herpes virus appears in the body of this individual. Since It is a protein from a virus. It is considered as a non-self to this patient or to this individual. Now, the immune system of the individual triggers an immune response against herpes virus characterized by the production of antibodies that bind to the envelope protein of the HSV. They neutralize this envelope, they eliminate this virus, and they create an immunological memory to the harmless virus, the herpes virus. So, who is the donor and who is the receiver of the transferred gene? The donor is the herpes virus. The receiver, we have the harmless virus, and we have the human being. What is the enzyme used to isolate the transfer gene? It's the restriction enzyme used to cut and to isolate the gene of interest. Why is this technique considered or qualified as transgenesis? Since we transfer a gene from one cell to another or from one species to another, and since the receiver expresses the gene. So here we transfer the gene from the herpes virus into another virus, then into a human being. Now the harmless virus and the human being begin to express the envelope protein of this virus. That's why this technique is considered as transgenesis. Here the correction of the exercise. The question is, what could be a potential problem when giving live vaccines? There is always the risk that the live viruses mutate back to their original harmful form and cause the disease. Knowing that the live virus vaccines are made up of attenuated viruses, but in some cases or sometimes we have a risk that the live virus returns to its harmful form, and thus it can cause the disease, and it can be fatal, leading to the death of the individual receiving the vaccine. But why are live vaccines more important than peptide vaccines? Because live vaccines, unlike peptide vaccines, produce a complete immune response to the virus, since we inject the whole virus and not only a part of the virus, and usually the immunity triggered against the whole virus lasts a lifetime, unlike the peptide vaccines that need several injections and boosting. Here we have a comparative table between peptide vaccines and live vaccines. For the advantages, the peptide vaccine has no risk of infection. The disadvantages of the peptide vaccine, they trigger a limited number of immune responses. Concerning the advantages of live vaccines, they produce a complete immune response that lasts a lifetime. But for the disadvantages, we have always the risk of mutation of the live virus to its original harmful form, and it can cause the disease. Hope you have understood the activity. Study well, my AS students, and thanks for listening.